Okay,
Okay.
great. Thank you so much for having me. Thanks for the invite. I always love participating in NeuroHackademy. I think it's a really fun format of meeting, and the fact that there's these lectures as well as the interactive tutorials and the hack sessions, I think has helped me learn a lot, in addition to, hopefully helping others learn some new things as well. And so, as Noah said, I'm going to be talking about pre-processing intracranial EEG data with MNE-python. And so before I start, I wonder if I can kind of see the people in person, and then I can see some folks on Zoom. If you're on Zoom, could you use the raise hand function if you yourself use intracranial EEG in your work? And if you're in person, could I see raised hands? And then maybe somebody can tell me how many folks there are in that. I can't see. Are there any?
Yeah, there's a handful, maybe half a dozen, maybe.
Okay, okay. Great. All right, wonderful. And how many of you are already using MNE-python?
Looks like we maybe have one in the audience.
Okay, okay, great. So that's helpful. And I think one thing that I wanted to kind of talk about here is how this is similar or different to looking at other types of data. I don't have a direct comparison. Actually, in my previous, I think, NeuroHackademy 2020, I did do a comparison between scalp EEG and intracranial EEG, if you want to go back and look at those materials. So if you work with EEG or if you work with MEG, you may be used to thinking about the types of artifacts that you should worry about, how to know whether you have good looking data. And so I think both for the people who already are using intracranial EEG, as well as those who are interested in looking at it, I hope that I can provide you with some information about what to look for and also some of the caveats that come with data sharing with data where there's a lot of stuff going on in the room. And if you're not the person who's collecting the data, sometimes it's hard to know that. So let's get started. So in my lab, we study how the brain processes speech and language. And I really like this kind of pretty art. This was actually made for an article in Quanta magazine where I got interviewed about some of my recent work. And so if you look up Quanta magazine and my name, you can read this article, and they made some beautiful art. So I just like to put it in talks now. But I'm very interested in how the brain is able to take in acoustic information, like what you're doing right now, and transform that into meaningful language. And we use continuous stimuli for this. I think there are a number of people in the audience who maybe are also interested in naturalistic stimuli and how to work with continuous story listening data or movie watching data. I know Kendrick K is here and so has a lot of data sets on natural movie watching and things like that. And so this is something that I'm very interested in my lab and in my previous work. And so we are able to use a lot of the tools, including MNE, to get to the point to where we're able to look at things like which brain areas are responsive to which different features of speech. So this is not a new problem, of course. So trying to talk about where in the brain processes speech and language has been going on for quite some time. And so even in the 1870s, we were learning from lesion models about which areas are responsive to either being able to understand language. And we see a lot of that in the posterior superior temporal gyrus, and then areas in the frontal lobe for producing or planning and sequencing aspects of language. And I'm showing these, I know this is not particularly a data talk, this is going to be more of a tutorial, but I do think that when you're analyzing intracranial EEG data, it does help a lot to have some a priori knowledge of where you might expect to see activity based on your electrode coverage. And so I'm showing these because we will look at an example natural data set, and you can kind of look at the coverage of the electrodes across the brain and kind of see if these are going to be in areas that you think might respond to your feature of interest at all. Okay. So this process that I was talking about involves transforming acoustic vibrations into spectrotemporal information at the level of the cochlea, and then somehow combining that spectrotemporal information to create something more meaningful, and also something where you could have very different spectrotemporal information from two people who have very different voices, and maybe they're saying the same sentence. Like you could have someone with a very deep voice versus someone with a very high-pitched voice, and they could say the same sentence. You'll know that it's the same words, but at the level of cochlea, you're not going to see that. And so with some of these data sets, if they include additional information with annotations for these different events, then this can help you to be able to look at encoding of specific aspects of sensory processing, whether that's speech and language or whether you're interested in something completely different. Okay, so this is thought of as a bottom-up process and involves kind of the whole auditory pathway. But for a lot of intracranial recordings, you're mostly going to be getting coverage of areas in the lateral surface of the brain if it's electrocorticography, which I'll go into in a second. And you may also get depth electrodes that are going to go through the cerebral cortex and potentially hit gray matter areas all over the brain. It really depends on where they're needed clinically. So as I said before, and I think some of you in the audience work with this type of data, but intracranial recordings are typically used clinically for seizure localization. And so these are placed by a team of surgeons with lots of planning from an epilepsy team, including epileptologists, neuropsychologists, the whole bit, neurologists, neurosurgeons. And the point of these recordings is to record from brain areas over the course of maybe 1 to 2 weeks, typically. And then during that time, you would have electrodes in different areas, as is shown here. So each of these kind of white dots is an electrode. It's overlaid on top of an MRI scan. And here it's shown kind of in 3D for this person. This reconstruction is not great, as you can see, and that's also something that you'll probably notice if you work with some intracranial data, is that often those MRI scans are, they may have other lesions or things that you have to deal with. That's a topic for another day. In any case, from each of these electrodes, you will be recording the electrical signals from these electrode channels. And that's going to record the local field potential at each of these channels. And in my lab, we process these in a particular way, and I'm going to share with you some of the code that we use to do that. But this is just showing an example of some raw electrocorticography or IEEG data. And this is collected throughout those 1 to 2 weeks. So that's the clinical aspect, is we are, we have electrodes in the brain, and the clinical team is waiting for waiting for seizures to occur. But seizures are not happening all the time. And so that's where researchers like myself come in, and we go and we ask those patients whether they would be willing to participate in research during their stay in the hospital. And so what this looks like, and this is a paper by Kai Miller, and includes some, I think, data release as well from Nature of Human Behavior in 2019. And they try and show what this looks like a little bit. I'm not sure actually whether this is an actual patient, because these look like jeans that he's wearing. Maybe they're pajamas, but I was thinking, a lot of the time they don't wear other things under the gown. So I'm not sure, it could be staged. But in any case, this looks very much like what this setup does look like for research. So you would have your patient participant, They're going to be in a hospital bed, in the hospital room. There are a few groups across the world that have specific lab spaces that they would move a participant into to do recordings, but typically that is not the case. That is quite rare. I would say for the most part, they're in their hospital room, they're in their hospital bed, and there's lots of other stuff in that room. So we bring in things like a recording system that might take a copy of the electrical signals that are coming off of the implanted electrodes. And typically you don't really see those implanted electrodes because there will be this kind of turban bandage that's hiding everything, and the wires are going to come out of the top of the head. And then you'll have some sort of setup where you can present stimuli to your participant while you are also recording neural activity synchronized at the same time. And so this is what we do in my lab. This is what a number of sort of sensory processing labs do. There's also cognitive tasks. And so a lot of this is thinking about also how to align that neural data to the events of interest. And so for some data sets, you may see that they include the actual stimulus that was played. For some, there may be annotations of the timing of, when is each trial? If it's a sentence, when does that sentence start and stop? Some groups have trigger pulses that they use to align the data. Some use a photodiode, which measures the luminance of the screen that has visual information. So I think in some ways, for those of you using the fMRI or EEG, the synchronizing is somewhat similar, but I think it also varies a lot from lab to lab. And this has been a big challenge in terms of data sharing and standardization, is just that every group has their own different setup. And it's kind of the Wild West of recordings. And so I think, sometimes there's some frustration about, well, why isn't there more shared? And it's because no one's stuff is in the same format. Not in the same format. They're not syncing it the same way. And so I think people are making efforts to standardize. And the bids, which I think you've already heard about, is one way of thinking about that. There are also other formats that people have proposed, but I think I would say IEEG is still much worse and earlier than the other neural recording methods. I think fMRI is probably the most standardized in terms of what I've seen from other groups. So before I go on, any questions about any of this setup or the context in which we're doing this?
Just out of interest, do you guys normally put up any sort of like behavioral information or background information when you share that data, or have you been in the process of sharing? Or?
So I haven't shared any data yet from my lab because we're still in the middle of collecting it, but I do think that sort of thing is kind of necessary. And actually when we collect data, we have trial notes where it's, and I'll get to this in another slide, it's even things like what else is happening in the room. I mean, there's certainly things like behaviors like button presses that, yes, absolutely would go into some kind of event file. And so you would be able to have those, or they're % correct on some behavioral measure. But do you mean more like
the clinical background and comorbidities? And
yeah. So I haven't seen as much of that. I think it's limited, but I also think that is relevant. We've been in my lab collecting, we have neuropsych data on everyone, we have demographic information, we have so it's a number of different cognitive batteries, and for verbal and nonverbal memory, IQ, motor function, all of those things. But I haven't seen as much of that. So I don't know if others have, but I agree that's an important thing to think about. Anyone else? Okay, great. Well, feel free to interrupt me. So in thinking about how the signals differ between different types of recordings that you might come across, I just wanted to show a couple of things. So this is a figure from a new a very large consortium paper out in NeuroImage just this year. And I think now the second round of proofs is up. And so you can go to NeuroImage and check out this actually very long article. It's over 60 pages of PDF, but it's a best practices for intracranial EEG. And so it really gives a very nice overview, I think, of all of the different considerations of working with patients, how people analyze their data, how they record their data, ethics, all of this, how they visualize data. And so you may come across data that includes either stereo EEG electrodes, and what these are is they are depth electrodes, and I think somewhat confusingly named because when I see SEEG, you might think scalp EEG. These are invasive. So they are not the same as scalp EEG. These are actually inserted into the brain. And these are used in cases where the seizure onset zone is hypothesized to be in a deeper source. It's actually also replacing electrocorticography, which are the surface grid electrodes that you may also see in this data set that I'm going to show you. But depending on the group, there are, I guess, preferences between the different groups over which of these works better for seizure localization. I know in a lot of European groups, I think they are mostly doing only SEEG at this point, and grids are becoming more rare, partially because of the need for a much more invasive surgery for grids, which requires a full craniotomy in a lot of cases where a section, a flap of bone is removed. Whereas for these SEEG electrodes, it's a burr hole. So that's actually, it still sounds, not great, but it is a lot less invasive comparatively. However, when you're recording from these, what you're able to get is essentially a similar signal to what you would get with EEG. You're recording voltages that are representing the summed postsynaptic potentials of the neurons in the brain, but at much higher SNR than EEG. So this is nice for a lot of reasons because you can get a lot more out of fewer repetitions of your stimulus. So you can do things in a shorter period of time. And also you can really localize where your signals are coming from nicely. And just to compare the sort of signal resolution in space and in time, there are lots of versions of this particular diagram, but here I would say ECOG in terms of temporal resolution, we're at the millisecond level. So unlike fMRI, where we're talking about bold signal, which evolves relatively slowly, ECOG is much faster. And the spatial scale, I would say, is maybe similar to fMRI. I'd still say fMRI is probably a little better, and also much better. fMRI is much better in terms of overall spatial coverage. So one thing I also wanted to discuss is that when we are looking at these neural signals, let me go back here. You can look at the raw voltages and you can bandpass filter them into different bands, much like you do with EEG. So you could bandpass filter and look at theta oscillations or alpha oscillations or whatever is your band of interest. You can also create event-related potentials, much like one would with EEG. So time locking some signal to a given event. In my lab and in my postdoc work in Eddie Chang's lab, we mostly looked at one particular band in the 70 to 150 hertz range. And this is referred to in the literature as high gamma. And we know from animal work that this is actually correlated with multi-unit neural firing. So it seems to be relatively precise in space and in time. And if you look at, for example, a frequency decomposition of your IEEG signal, this is from one electrode, but this is actually averaged across 10 repetitions of this sentence. It had gone like clockwork. And you can see that in this 70 to 150 hertz band, the increase in power is shown in red. And there's this pretty kind of punctate time-locked activity that you can see in this range. You can, of course, also see other oscillations. We could have plotted this even lower if we wanted to look at delta band. In this case, our sort of default settings for some of my old processing code went down to 4 Hertz. So there's no particular reason that we cut it off there, other than this was just, we're more interested in this range. Okay, so in my tutorial, I'm going to be showing you how we extract this high gamma band range and then create some plots with it. Okay, so as I said before, I think a couple of the advantages of intracranial recordings are the fact that it can measure changes at such fast timescales. So if you want to know things like the difference between a B and a P sound in the brain, that's only tens of milliseconds that gives you actually the ability to resolve those two sounds. So knowing that I said the word bear versus the word pear, that's a very sort of very fast difference. And so you would be able to do something like that with intracranial EEG. And you also tend to know where your signals are coming from because they're directly in the brain tissue. And if you're bandpass filtering in this high gamma range, it's from one electrode to another, you can often see quite different responses on those electrodes. And then finally, as I said before, it does have very high SNR and it requires fewer trials than something like EEG or MEG. So just as an example, this is an evoked kind of heat map of some single trial high gamma responses to the syllable saw. And if you look just at these 5 repetitions on one electrode, they're very similar to one another. So if you're used to looking at, I'm also doing some scalp EEG in my lab. If you were looking at the response to saw in EEG, you have to average over a lot of repetitions of that to get your nice evoked waveform. But here, it's beautiful. You can get it in very few repetitions. So this is very nice. So, okay, beautiful. IEG is wonderful. Love it. But there are also disadvantages. So as I said before, a lot of the coverage depends on clinical need and is not based on your research question necessarily. Unless maybe your research is about seizure onset zones, and then that's great because you probably would get great coverage of the areas that you need. But for me, I sort of, when we're talking about recruiting patients to studies, we sort of work with what we get. And so they may have coverage over temporal lobe areas for language, they may not. And so then we can change the tasks that we do. And you'll see also with the shared data that I'm going to show you from Nick Ramsey's group, there they have many, many different participants, but all of them have very different coverage. Some of them have coverage over motor areas, some over temporal areas, some occipital. And so if you're trying to pool across participants, you should not do this blindly. You definitely have to go in and look at the data and see, where do I have electrodes? What are the regions of interest that I have good coverage over, and what do I expect to see? Of course, another couple of things that are disadvantages are that these are invasive and have to be done in patient populations. So if you are interested in sensory encoding and not necessarily epilepsy per se, pretty much all of these recordings are coming from patients who have something where they needed these electrodes placed for clinical reasons, whether that was because they're having seizures due to epilepsy or a lesion, they may have a tumor. And so that can also affect the network. And so being aware of that, I think, is important. And so I think Jennifer's question about that, I think, is important, because if you don't know where the seizure onset zone is for someone, then how do you know how to put your findings into context when you're actually trying to interpret what you get? And then finally, it's a good thing that the SNR is good because the experiments are often quite time limited. It's very difficult to design a task that, well, maybe it's not difficult to design the task that's very long, but it would be difficult to execute your typical psychophysics or psychoacoustics task where someone has to sit there for a full hour or an hour and a half making some decision. So in my lab, I sort of insist that all of the tasks can be done in 5 to 10 minute chunks where they can have breaks in between, or even if they just want to do 10 minutes and then we come back in a few hours, then that's fine too. Because these are folks who are in the hospital, they may be having a hard time. We can't push them. It's not appropriate. And I think it's also important to acknowledge that they're going through something that is difficult. I will say in some cases, people like participating in the tasks and they find it to be a way to pass the time, which is also nice for us. If they don't like that, if they don't like the tasks, then we just typically don't get any data or we don't get as much data. The other thing that I wanted to bring up, which I think is important for data sharing, and I don't know how we should share this, and I would love to hear from you all what you think about this, but there are a lot of things in the room that are not as controlled as your typical research environment. So for example, there's all these background sounds that can happen all the time. The infusion pump, for example, for giving them medication. This makes a beeping sound that is like, can be just constant, and you can press the silence button and it will silence it for a little while and then it comes back on. And so just the number of sort of beeps and boops and different medical equipment that you can't turn off and you should not turn off. This is not something that I think is always recorded. And in my lab, we do have microphone recordings of everything that's going on in the room, but then this can also be a privacy issue if you want to share that, because if you have any private conversations that are happening or they happen to say their name, you have to kind of redact information from that. And so this is something I'm kind of trying to think about, and I'd love to hear from others about how to best do this. You could also just think of it as, well, it's just another source of noise, and as long as it's not correlated from trial to trial, then kind of whatever. And I hope that the people who are collecting the data try their best not to have too much background noise or to minimize it to the extent that they can. But if you're not there, you can never know whether someone was having a conversation in the corner, whether there was a nurse that came in and took blood pressure, 4 minutes into your trial, because these things happen, right? So you get interrupted all the time, even if you sort of try to mitigate those things. And so this is, I think, a very different research environment. One more thing, sorry, to say about background sounds. A lot of the patients also have these calf pumping devices that are meant to prevent blood clots, but they make quite a bit of noise, both in terms of their sound and also they can make electrical noise. And same thing with some of the electronics around the bed. So the people who are collecting the data, I think, have to make a big effort to kind of minimize these sources of noise. But I think also if you're coming into this naively and looking at shared data, you kind of should be aware that sometimes if you see something weird in your data, it might not be a brain thing at all. It could be something else going on in the room. And so I think thinking of ways for annotating those sorts of issues and actually sharing that would be very, very helpful. Okay, and then as I said, also thinking about where the electrodes are located is important, how they are plugged into the amplifier. So in some cases, I think the researchers will share this, but there are many different recording systems. And so part of what you do when you are pre-processing intracranial data is you may re-reference the data. And the way that you choose that reference often depends on where the electrodes are located, as well as if you know if some of them are all plugged into one system, then maybe they'll have, or if they all have one cable that's shared, then maybe that's a shared source of noise. And if they're plugged in banks of 64 channels at a time, then this is also something to know. So these are other things. And then, as I said, sources of electrical noise. So making sure that any sort of other equipment that might be interfering is those sort of noise risks are mitigated. I'll tell you the funniest example of a source of noise was I was working with a patient where she was holding hands with her boyfriend, and her boyfriend was holding his phone, and his phone was plugged into the wall. Every time they held hands, we would see 60 hertz noise, electrical noise in her brain signals, but only when they were holding hands. And so then we felt bad because we said, sorry, can you please stop holding hands or unplug your phone because this is, causing noise in the data. So just all sorts of, I think, kind of weird stuff that you have to think about when you're looking at these data. And so I think that's also been maybe a source of consternation for the people who are trying to share it as well, is sharing that information is tricky too. Okay, and then finally, thinking about acquisition sampling rate is important if you want to be able to look at high frequency activity, like what I was talking about with the high gamma activity. Okay, any questions on this so far? Okay, great. All right, so let's talk a little bit about some of the IEEG artifacts that you might see. And this is not exhaustive, but certainly like other types of recordings, you can see motion artifacts. So motion is not your friend. It's not as bad as EEG for things like eye blinks and clenching. That actually is a lot less of a concern, at least in the recordings that I look at. I do believe, and I've been told by other researchers who look at electrodes that are maybe closer to the eye orbits, that sometimes you could get artifact from eye blinking, but you don't see the same kind of dips that you might see in EEG activity. You do see, if they move the cables, for example, from that turbine, sometimes people will shift in their bed and they move around, and then you'll see this big low frequency drift. You also may see epileptiform artifact. And so those are some things to kind of look for. I'll show an example of some of that, but it's also good to be able to have someone with clinical knowledge to speak with about these sorts of things, because you want to correct for some of these things, but like any data set, you don't really want to overcorrect. If you get really happy with rejecting every single piece of data that looks slightly like there's a little tick in it, then you will end up with no data and you won't be able to sort of see anything. And so I'll show you some that I think are more obvious that I would remove, but this is also kind of I would say an art in a way. Chewing and jaw artifacts you may see as well, especially for stereo EEG. If there's electrodes that are close to the surface of the head that are what's called near the bolt, then those you can see more myogenic artifact because they're close to the muscle. The deeper ones, you don't typically see it in there. So this is something to think about, because sometimes you may want to look at your electrode montage, which means how the electrodes are placed on the brain before you start pre-processing and referencing, because you can actually make those artifacts go into your other channels if you're not careful about the way that you're re-referencing and you don't look at them first. And then the last one that I think is something I hadn't seen before until recently is some folks with epilepsy have what's called a vagus nerve stimulator. And you can see what's a vagus nerve stimulator artifact. And that is going to be a repetitive stimulus that I will show you an example of in just a second. So let me show you a couple of examples of some seizure activity. So these are two examples from the same person. So this is a participant who was listening to and watching movie trailers while we recorded activity from a grid. And on the right-hand side, this is the sort of normal intracranial EEG activity on the first 20 channels. And then this is during a time where he started having seizure-like activity. And you can see this sort of rapid synchronization of all of those channels start to show up. Now, what I would do in this case, actually, this person did have a seizure during a task. And so we actually stopped the task in the middle and we marked that. And we are not using this data as, for our task analysis. Number one, because we had to stop the task in the middle, but also number two, all of this epileptiform artifact means that this person is not going to be processing those sounds in the same way as they would while they're not having a seizure. So you may see things like this, although if they're shared for research purposes during a sensory task, they may not include these. If they're sharing specifically clips of seizure activity, which some groups do, then you may see more things like this. With this synchronized activity, it could be on a subset of electrodes, or you could even see all of them synchronize together. And then this is another one that I find helpful to be able to localize, which is a spike wave discharge. And what this looks like, this is typical of certain types of epilepsy, where you will see a fast spike followed by a slow wave discharge. And they may occur during what are called these interictal periods, where there isn't a full-blown seizure, but maybe there are spikes every now and then that show up. They may be on a subset of channels, or they may spread to other channels. If they are in a convulsive seizure, you might see these across many channels, all synchronized and rhythmic. But if you see kind of one-off every now and then, one of these, then you can mark it as a bad channel, and I'll show you how to do that. And then the VNS artifact, as I was saying, for vagus nerve stimulation, this looks very interesting. So here's an example of some intracranial EEG activity. I'm showing a lot more channels here at a time, but you might notice that for some of these, it looks like there's kind of like rhythmic, just really short little spikes on some of these channels. It's not really all of them, but if you have a good eye, then you see there's this repetitive thing that's happening on RALTA 13 and 14, and you're seeing this. If you looked at the power spectrum of this is what that looked like. So this is repetitive sort of artifact. This is not neural activity for sure. It's also not the same as a line noise, like 60 hertz noise that you might get from an electrical artifact. This particular one was a 20 hertz signal that we were able to get rid of with a notch filter or with a common average reference. And so if you do come across things like that, you may have to think about how to analyze that data differently. You may want to kind of note that as well and potentially not include it in your other blocks. But I think this is something to be aware of as well. Okay, so now I'm gonna start the tutorial. And so I'm going to be running this on my own computer. I think there's also a version of this on the Jupyter Hub. But one thing I would say about that version is at the beginning of the tutorial, there's some stuff that requires the interactive component of MNE-python. And so being able to actually scroll through the activity is important. And so we're going to do kind of a more basic analysis with this. We're going to load some IEEG data with and without the BIDS metadata, because I want you to see what it looks like when that nice metadata is included versus when it's not, and for you to kind of be able to recognize what to do if you found some that doesn't have those annotations that you're kind of expecting. And we will plot the power spectrum, which is, what I showed over here with the VNS stimulation. Sorry, just lost my slides there. And then we will also convert to, we'll plot the raw data to look for bad channels and artifacts, convert to high gamma and plot some evoked data. Okay, so let me share a different, let me just share my whole screen instead. So, and while I do that, any other follow-up questions from anyone? Hopefully I haven't just scared everyone off of ever looking at intracranial data, but I do want to kind of talk about the things that people should know about and be aware of, because I think it is important to have that kind of context. Okay. Do you all see my screen? Yes. Okay, great. So this is our notebook, and we will be looking at data from a freely available intracranial EEG data set that has actually a lot of data from Nick Ramsey's lab. And Julia Berezutskaya is the person who curated this data set. And this is from a number of people who are listening to and watching a short audiovisual movie. And so they have a number of different participants. So here we have 63 participants. So it's actually this is a large curated data set. This is, I think, a lot larger than many of the data sets that I have seen. And some of these have intracranial EEG data and fMRI scans, or not fMRI, I'm sorry, and structural MRI scans. Some of them have just one or the other. And they were collected over the span of, I think, 10 years. And so this does take a long time to sort of get, but I am going to be working with the data from subject 6 for now. And so if you don't want to download that whole data set and you're trying to follow along on your own computer, then you can do that with the subject 6 data. And I'm looking at the stuff that's in the session IEMU, and we're going to look at this particular set of data. Okay, so, all right, so what we're going to start with is we have a number of loaders here where we're going to be using MNE. We're going to use some features from Matplotlib, NumPy, Pandas, OS, just to sort of do some data manipulation. And we're going to use MNE bids for some of the nice reading functionality that it has. You may need to pip install some of these things if you have not. I have also included a pre-processing utility script, which in my own folder looks like this. So this has some of the guts of the transformation to high gamma. And so we can talk about that if you're interested in the implementation of this as well. But this is what I use in my lab. I know that there are functions within MNE to do Hilbert transforms, and I'm actually not using those here, but we can talk about whether maybe this is something we should add to MNE-python, and someone can help me work on that next week. So, and then also here, I'm using the Matplotlib TK backend so that I will be able to plot interactively. So let's load that information. It pops up the Python window over here. And now I'm going to, you could change these variables. If you're working on the Jupyter Hub, you may be able to load some of the other files. There are some issues with some of the TSV files. So if you do have some issues, we can maybe talk about it offline for other participants. I think I know what's going on. So here, these are in bids format. So we specify the subject, the session, the task that they're doing. And in this case, there was a film task and a rest task, and acquisition and run are always clinical and run 1. And now here, this is going to be the path to the directory where your data are located. Now, actually, I have to change this again here because there we go. That would be your own data path to your bids folder. And so I'm going to now run this. And the nice thing is with this type of reader, the get bids path from fname, and getting bids data, we can use the read raw bids loader. And what this will do is it will extract a lot of nice information from the files within your bids data structure. And that's going to give you information about where the channels are, information about what the different events are that are related to tasks that the person is doing. It even has what the researchers identified as the bad channels, things like that. I'm also going to load another version of this, assuming that we just had the original data, which in this case, we have a vHeader file, and this is associated with a .eeg and a .vmrk file. This is the brain vision format. It's actually equivalent to, if any of you use EEG and brain vision, then this is the same loader that you would use for that. And so here we can just load that data and it loads just fine. But I'm going to show you the difference between the two. So now when we have loaded in that data, we can look at the information about this data set. The measurement date has been anonymized, so obviously it was not recorded in the 1900s. This is not like some, I don't know, very early Penfield. I mean, even that's not the right date. But this is just anonymized. This is the participant number. Then these are the good channels and their channel types. So this is very nice as a structure to be able to look at what kind of data are you dealing with. Here are the bad channels that they have marked. They don't have any EOG channels for ocular artifacts available, but they do have an electrocardiography. So for heart cardiac artifact, you could use that. And then the sampling frequency for these data is 512 Hertz, and they have not been filtered. So the high pass and low pass filter are just to show you whether there was already any kind of filtering that happened on the data. If this is half of the sampling rate, then essentially that is the Nyquist frequency, so this is not filtered. You should also probably look at the channel names, and these can give you some idea of where these channels are. But also, this is another thing with intracranial EEG, is that there's currently not any agreement on how to name IEEG channels. So it's not like EEG where you always have CZ, FC, you know where the temporal electrodes are, you know where the occipital ones are. Each group may have their own naming convention. And so this can be tricky as well if you're coming in without sort of knowing that context. Of course, you can look at the sort of screenshots of how they're plotted on the brain. And there are, at least in my group, some of these have names that are related to where the electrode enters the brain and where it exits. And so there are efforts within the clinical community to try and standardize this, but this is also still not standard. So, okay, so let's first look at the power spectrum for these recordings. And so this is the first thing that I do. After I've loaded the data, I always like to look at, what does my power spectrum look like? How noisy are the channels? Have enough bad channels been rejected? What does this look like? So I'll run this. And if you're running it in Jupyter Hub, you may just see it in the Jupyter Hub output itself. But if you have an interactive window, then you'll see something like this. And so note that these have been colored according to the electrode that is being displayed. And they're shown on this head model, which is not as useful for intracranial data because you don't see where it is in the brain, but at least you see from the top surface, these ones that are more in blue are closer to the back. The ones that are in green are closer to the front. There's also some depth electrode kind of in this goldish orange. And you can click on these and you can see where those are. You can also see that there's one over here that's got some kind of high frequency drift over here. They haven't marked this as bad, but you could also when you look at the raw data, kind of figure out whether there looks like there's anything strange with that electrode as well. So what I would like to look at here is look for kind of outliers and see, are there things that look particularly different from the rest of the channels? So this looks pretty good. What I want to compare this to is the one without the bids metadata. So let me run that. And what you'll see already is, look, there's no colors and it looks very confusing. If you have never looked at these before, it's like, what is this one? And then you can click on it and you see, oh, it's MKR1+. Okay, this is probably a marker. And so this is likely just an oscillator or something like that that's helping them to synchronize the activity. So this, in this case, these electrodes have all been loaded as if they were EEG channels, even though they probably are not. And so you could look at your channels this way. And then what I like to do is, the next thing I'll show you is just plotting them as a kind of time series. And then you could reject them and then replot the power spectrum to see how that changes and whether you have kind of gotten rid of adequate numbers of electrodes. So I'm just clicking on some other ones that are high up as well. And it looks like that's R3+, T03, P41. There's another oscillatory one, Abdo plus, so some abdomen electrode. And so I'm going to close that, but this is what this can look like if you are just working with kind of not annotated data. So if we do then want to plot the data and look for bad time segments, then we can use this feature, which is raw.plot. And I've added a bunch of keyword arguments to this. You could just do raw.plot like that, and this would work just as well, although the scalings are often slightly off. And so I like to do scalings, auto. I color the EEG and ECOG electrodes blue. Sometimes they're called EEG, sometimes they're called ECOG, depending on how it's stored and who made the file. And then how many channels do you want to see at a time? And blocking is just whether you want it to return your terminal window or not. So in this case, this doesn't really matter, but I'll run this anyway. And so here, this is the part that doesn't work so well with a Jupyter notebook because these are interactive. And so you can see here, we've got our information kind of organized by different channel types. The ones that have been marked as bad by the researchers show up in gray, and you can actually click and unclick them if you want to select bad channels or to add to your bad channels list. And you don't have to do anything to save them. They just get saved automatically to that raw structure, which you are then later manipulating. And so remember how before, when we were looking at some of those ones that had a really strange power spectrum, they were things like this marker 1 and marker 2. And you can see here that these are just kind of square wave pulses that are being included probably as a method of synchronizing these data. I can also scroll up and down, and I can scroll to the right and kind of move through this activity. And here another nice thing is that we have these annotations that have been pulled in by the bid structure that allow us to see that this was a blocked design where they're playing music. And then during the green parts, they are playing speech. And so you can kind of see which data aligns with which part of the trial. And so this is really nicely annotated. But I will also point out, so a few of the things that we were looking at earlier were things like spike waveform discharges. And so already here, I see something that looks suspicious to me. And so what you can do is, let's press the help over here. So we can see here, these are all of the things that we can do with this scrolling window. You can scroll left, right, you can add more channels, you can decrease or increase the amplitude. And so if I decrease the amplitude, then you can start to see whether some of these channels are indeed having these large amplitude spike wave discharges. And so that's what I see here. And for something like this, if they're not happening that often, what I would probably do when pre-processing this block is I would go into annotation mode, which is you can toggle that using the A key. And then this will pop up a window, and then we could add a new label for bads. And so I'm just going to do that. And then we can click and drag. Oops. We can click and drag, and it should allow us to add those bad times. And for a lot of these, I like to try and encompass the whole time period. So if there are two that happened one after the other, you are going to want to just mark all the way through it, because there's honestly not very much point to looking at the very tiny in-between data that you might get here. And for me, what we do is we actually remove these time points entirely. I know that in EEG, there are ways to remove, for example, blinks and other artifacts through ICA. I'm not sure I feel great about that, because if there are sort of instances of seizure activity, they could be affecting your network in other ways. So I would probably err on the side of dropping those time points. Okay. All right, so I know it's almost 11 a.m. So how are we ending at the hour, or do I have a little bit of time?
Yeah, so we have a coffee break at 11, but if you go a little over, that's fine.
Okay, all right, I'll try and go sort of quickly through the rest of this, and also just compare. I do want to compare this sort of bids and no bids really fast, and then we'll talk about referencing and a little bit about high gamma. So here it is. If I just loaded it in without any of that bids information, it still doesn't look terrible, because actually the speech and music time points have been saved in the file, but you will see the order of the channels is different. And there are things like this marker occurs in the middle of the file, and all of these other miscellaneous trials are also kind of in a strange order. They're included as EEG channels. And that's also why that power spectrum looks so funny when you plot it, is you're plotting a lot of things that are not brain activity. So overall, I think using those types of readers is very helpful. And so storing the data like that is helpful. But if you do encounter things where you see a channel like this, for example, this is very likely not a neural channel. And then this one, this really fuzzy one that you see here, this is a good example of a bad channel. And what I mean by bad is often the electrode contact might not be good with the brain physically. It also could be that it's not plugged in very well. Maybe that, so each of these has to be plugged in individually to an amplifier. And if it's slightly pulled out of its socket or something like that, or there's any damage to the wire, then you can get a bad electrode. So it doesn't necessarily mean there's anything wrong with that brain area. It could be not the patient's fault at all. It's just that you didn't have good electrical signal. So those are some things to look out for. Okay. So now this is the part where I think that this could make for a good hack project. So referencing your data is something that you really need to do in order to kind of get rid of shared sources of noise. And you can do typically 3 different things. You can reference your data by subtracting the activity from a single electrode from all other electrodes. You could base it on the average of all the electrodes. This is often called a common average reference, or CAR. And this is actually what I use for most of my recordings, but it's not a good idea if you have only electrodes that are in one small area, because if you're subtracting the average, you're going to remove a lot of signal in that case. And so you only really want to do it if you have widespread coverage of areas that are not functionally doing the same thing. Then the CAR is a good idea, because whatever shared signal these guys have. Whatever they're sharing in their electrical signals is more likely to be some sort of external noise. And then the final one is bipolar referencing. And this is where you have pairs of electrodes that are next to each other, and you subtract one from the other in order to form a kind of pseudo electrode. And this allows you to look at more local signals. However, this is this has to be based on knowledge of where the electrodes are. So let me show you really quick the difference between single referencing and common average. So we'll plot those two, and this is going to be referencing to just channel P0. And so what it's going to do is P0 is now flat because you subtracted it from itself. It's now the reference electrode. And now everything else is relative to P0. And I'm just going to close this so we can have an example of what it looks like if we were to do a common average reference instead, which is using this set EEG reference and then plotting it. And so here you don't see any flat electrodes. And in fact, just looking at this, it might be hard for you to see a difference between that and the other one because they're all just squiggles. But I'm going to show you a couple of things. So if we open this in a new tab and this in a new tab, then you can see the difference between, let's see if this works. So if I flip back and forth between the two, you can kind of see some similarities and some differences. This is the one with the one single reference, and you'll see that there's sometimes these shared peaks across all of the channels where there's just like a blip that happens in all of them, and it's synchronized across all of your channels. But then with the common average reference, that goes away. So I'll flip back and forth again. So see how there's like a peak, and now there's no peak. And so this type of reference is actually kind of my preferred reference. But I will say in the IEEG community, bipolar referencing is also very common clinically. And this would be, instead of having these electrodes like you see here, instead of having electrode 1 and 2 be separate, you would do 2 minus 1 to get the signal in between 2 and 1, and then 3 minus 2 to get in between here and here, 4 minus 3, etc. But the thing that's tricky here is that setup of which electrodes are you subtracting from which is going to depend very heavily on where they are in space. So you can't just say 2 minus 1, 3 minus 2, 4 minus 3. You could do that, but then once you get to 9 minus 8, 9 minus 8 does not make sense as a subtraction to do because they are not close to each other. That would give you some weird dipole right here. And so you want to be able to use the configuration of the electrode grid itself in order to kind of best choose the pairs that should be used in your bipolar referencing. So I think for now I'm going to skip that, but if anybody is interested in trying to figure out how to make a better bipolar referencing scheme for arbitrary IEEG, I'm happy to talk about that. At the moment, MNE has this way of setting the bipolar reference, but you have to give all of the pairs as lists. So you could do P1 and P2, subtracting P2 and P3, but there's no way to, as I see it right now, to use the information about where they are to kind of make that work better. Okay. The last part of this is the high gamma transformation. And so this is actually using, I'll just start it right now. It's using a separate function called transform data, where there are a number of different kind of options that you can choose here, which include whether you want to perform a notch filter, which I probably did not need to do because these look like there's no 60 hertz noise. They were also recorded in Europe, so that would not be the frequency. You would use 50 and 100, and even then I don't think it really needs it. But notch filter, and it pops up what happens with the data after the notch filter. Typically, if you didn't need a notch filter, this will look the same as before. If it was really fuzzy, then it will look much better. So this, again, depends on the original data quality and whether there was a lot of background noise or not. And now this shows after the common average reference, which we already looked at before, and we saw it gets rid of some of those really synchronous kind of blips. Close that. And now this one takes a little while. And if I go back here, it is going to be applying the Hilbert transform in an 8-band setup. And what this will do is it allows you to collapse across those 8 bands in order to get a higher signal to noise estimate of the high gamma activity. So this is what I'm calculating right now, is the power in 8 different bands from 70 to 150, as I'm showing here on the slides. All right. So I think maybe I will end with just showing you, after this is done, I'll show you how to plot the events, and then we'll call it. But if anybody has any questions, actually now while this is running, I'm happy to answer them.
Yeah, we have a question here.
Go ahead.
So I have 2 questions actually. Do you do any interpolation with the bad channels, or you just remove them? And the other question is, when you do the time frequency analysis, I know you have like blocks of 20 seconds, I think. Is that do you have any like do you have to do like any segmentations with the within the block, or you do like for the whole block?
So
Yeah,
that's a great
so
question.
thank you.
Yeah, so for your for the second question, let me answer the second question first. So for this Hilbert transform, we are doing this on the entire data set. It's actually better to do it on non-epoched data, because you do sometimes get edge artifacts if you are doing it on already segmented data. So even though we see the segments in the raw file, the thing that is being passed to this to this function is the raw data itself. And so that would be continuous. And so I think that works the best. And then your other question about interpolating channels, I typically do not interpolate between the channels because in this case, they can be different enough. From what I've seen from good channels, I don't know that it's a great assumption that you would be able to interpolate over the bad channels like you would for EEG. So I do not. Okay. So now it has popped up a window of what the high gamma activity looks like. And this is actually very different from what we saw before, right? So this looks quite different from the original intracranial EEG signal. And I will tell you, just based on my own kind of knowledge of what this looks like, this looks normal for high gamma. This is now an envelope of a signal. And so you are going to expect to see more kind of slow moving activity and less of the kind of fast oscillations that we saw before. You won't see anything that looks like spike wave, although let's see if we get to, and even there, it's actually sometimes tricky to see those during that time. And so that's why I also recommend doing the artifact rejection on the raw data rather than on the high gamma data. I personally try not to reject anything again after I have calculated the high gamma data. So I want to do everything on the raw first, because otherwise it gets a little bit circular, I feel. And then I worry about kind of trying to reject things based on what I think something should look like in my final signal. And so I personally don't like doing that, but you may see sometimes, not in this data set, but in some data sets, if people have myogenic artifact on especially SEEG channels that are close to the skull, once those get filtered out, that might look like a big high gamma signal, and it's not. And so you kind of have to be careful when you're looking through some of the data, especially at the channels that are more that are closer to the skull, and potentially remove those signals before you do the CAR and before you calculate high gamma. So that's what I would say about those. Okay. And so now the last thing we will do is we can load the events and turn them into evoked plots. And so this has a TSV file that we can load with pandas, and you can see the onset, duration, and trial type here. And what you could do is you could take this TSV file and convert it into something that MNE is able to use, because MNE likes these things to be in samples and not in seconds. And so here I have done that over here by just creating a set of lists and then zipping them together to create my set of events. But this is actually totally not necessary. For this particular event file, you can actually read them very nicely from the annotations. So if you didn't want to do it from your tab separated file, you could also just use MNE events from annotations, and that gives you the same answer. And actually here, I guess I did not need to multiply it by the sampling rate. So here, these should be the same.
Liberty,
Yes.
I really hate to have to cut you off, but we need to make sure we have enough time for our next speaker to set up.
Yes. Okay, no problem. So that's it. And basically, if you want to go through the rest of this, you can plot the evoked data and move through this, and it will show you how these look on each channel. For IEEG, I'd always look at it on the single channel level rather than averages, because you are going to expect that your individual channels are going to have different receptive fields, different sensory coding properties. And so in this case, averaging is not going to help you as much as you might think. So thank you very much. And if you have any other questions, I'll be there next week and we can talk in person and go through the rest of this. But thanks again for having me.
Okay, thank you very much, Liberty, for that great talk. For everyone else, we're going to have a coffee break now. We are going to extend the coffee break a little bit since we went a little long. So we'll be back here at 1:45. And we'll see you all then. Let's go ahead and thank our speaker again. And as a reminder, Liberty will be here next week, so you can talk to her then. So thank you again, Liberty.
Thank you. And I can answer questions in Slack too. Thanks all.